Okay, so our first speaker is Professor Mamie Coats from Alabama State University. She's been our collaborator for Act 2. And she's going to talk about the title here. Microbial competition between mucoid bacterial species.
Well, good afternoon, everyone. I am Mamie Coats from Alabama State University, and I would like to start off with a bit of a disclaimer. I'm a microbiologist, sorry. It's just what I do. And as I was reading and getting ready for my presentation, I was watching The Big Bang Theory, and I was just laughing at the way they're always, the more physical science guys are always laughing at the biological people, and then it flashed to me, you know that you're the biology person, right? So hopefully we won't have any Big Bang moments today, but I did change my topic from what I had mentioned earlier to Zaria, and I'm going to talk about how mucoidy affects pneumococcal infection in cystic fibrosis patients. So just a bit of an outline. I'm going to tell you the bacteria that I work with, and as well as talk about a collection of bacteria that we were able to put together, how this relates to cystic fibrosis, and how the bacteria behave in vitro and in vivo, and then open it up for any discussions. So in particular, this work focuses on Streptococcus pneumoniae, which is a Diplococcus, is gram-positive. I'm going to talk today mostly about the capsule polysaccharide. And what this is, it's just a sugar coating that's on top of the bacteria. But it's the main virulence determinant in the bacteria. It's what lets the bacteria live inside people. And this is going to influence what's called the serotype of the organism. The bacteria, most of us in here today, at least 90-95% of us are going to have Streptococcus in your nose. And as long as it stays in your nose, in adults and children, it's going to be fine. It's when the bacteria moves from being in a carriage state to a disease state when it goes into normally sterile sites, such as the lungs when it causes pneumonia, the middle ear where it'll cause otitis, or into bone and joints where it can cause bacteremia, septicemia, and arthritis. And while the bacteria is normally harmless, it is potentially fatal in adults and even worse on children. To the capsule. Right now there are more than 95 different types within this one bacterial species. You have 95 different capsule polysaccharide types. And the way to think about this is almost like they're siblings. Two parents have a set of children, but they're all individuals. And that's the best way to think about this bacteria. Not all the serotypes are going to behave equally. You know, everyone has that one kid that won't do right. Think about it like that. So they're not all equal. They don't all cause disease equally. They don't work the same way in animal models. 85% of the infection is normally due to about 23 to 30 serotypes. They're 95, but most of the infection is due to a small number of those. And classically, we're talking about type 3, which I'm going to keep using the word mucoid and mucoidy. And typically, that's going to be with serogroup 3, and it makes a large amount of this capsule, which even changes the way the bacteria look to the naked eye. But I'll go into that in a little bit more detail. And that sequence there, that's the repeating unit for the type 3 capsule. So what do we do? In 2002, Dr. Marilyn Crane at the University of Alabama in Birmingham began a collection of all of the pneumococci that came through Children's Hospital. Why did she do this? There was a vaccine that was instituted at that time called PCV7. If you have children or you follow the pediatric recommended cycle of vaccination in the U.S., you may have heard of PCV7 or PCV13 because it is part of that recommended scheme. Well, we knew that when you take a vaccine and put it into a population, you're probably going to wipe out whatever's included in the vaccine. But what most people don't think of is what's going to replace the bacteria that you wipe out. You're not going to make less bacteria in your nose. No, it'll change to something else, or other bacteria will take its place. Think about the seats in this room. If every seat in this room had to always be filled, but there was a line of people lying down the steps. So when one leaves, someone else just comes in and takes the seat. And that's what we wanted to watch. Also, the bacteria can change. New serotypes are going to come in that weren't included in the vaccine. And while only about 30 really cause disease, it doesn't mean that the other ones can't. It just means that they're not able to fight to get a way in to cause disease yet. So that's why the collection began. And as I came into the Dr. Crane's lab, I noticed that we had a lot of isolates from cystic fibrosis patients, which is unusual. If we're looking at all of the children that come through one of the major children's hospitals in the southern United States, I shouldn't see that many cystic fibrosis patients because they're a relatively small part of the population, but I did. So that made me curious as to why we were seeing that so often. And just a little bit on cystic fibrosis, it is genetically inherited. There's a mutation in the CFTR, which is located on chromosome 7. We mainly hear about cystic fibrosis and how it affects the lungs, and we think about lung infection, but a lot of the physical illness actually comes from the digestive system as well. What causes the lung infection in cystic fibrosis patients? Normally what happens in our normal pathway is that the salt and water balance is such that a cilia within our lungs are able to beat. And that ciliary beat is going to keep the bacteria from being able to adhere and cause infection. Well, in CF patients, what happens is that the mucus covers those cilia, so they're not able to beat. And it's also a rich environment, rich in nutrients for microorganisms, which is how they get infection. So what I found that when I looked at the particular bacteria that we were getting from CF patients, they didn't even look like the bacteria that I got from other children. They tended to be more highly mucoid. And that just means a glossy appearance on TSA blood agar, and they're usually larger, and they're much harder to manipulate. I don't know. We're not going to try that. But the ones, if you look at this slide here, that's considered highly mucoid, whereas this is considered a normal looking bacteria. See, they're much larger, like this is one colony here, and down here, this would be one colony. So they're going to be much larger, and they're different to look at, and they're actually much harder to manipulate. So how do these isolates differ, other than just in appearance? So we did capsular serotyping, and I'm not going to go through how this is done, but if anyone's interested, I'll be happy to explain it later. And what we found was that when we compared CF patients to non-CF patients of the same age, all of our strains are from children, we found that certain serotypes predominated. In this serogroup 3, which is the one I mentioned earlier, that tended to produce more capsule anyway, was statistically higher in our CF patients than in our non-CF patients. So the goal then became to see what does it have to do with cystic fibrosis. There are also some other isolates that predominated, but today I'm just going to focus on the serogroup 3. And I will say right here, this is one of the things with students where when you start to look for one thing, 15 other things are going to come up. We didn't start the project to look at cystic fibrosis. We didn't start the project to look at mucoid bacteria, but that's where we're going to go. So what does this have to do with the price of sweet tea in Alabama? And yes, I use my accent for that. Really, does the way these bacteria look have anything to do with cystic fibrosis? And to do this, we were able to use a mouse model that actually expresses the human CFTR gene in its lung. But this is a later generation cystic fibrosis mouse, so it doesn't have the intestinal issues that the earlier generations had. Typically, cystic fibrosis mouse models, the animals were very, what we would call puny. They didn't survive well. They might have lived a few weeks, but they died from intestinal problems. But this is called a gut-corrected CF model. So when we looked at these, we actually found something very interesting. And this is a strain table here. CHB means it came from our study. W2 is kind of the generic S. pneumoniae that most people use if you're just studying the bacterium. So it's not a recent isolate, it is a human isolate, but it was isolated many, many years ago. CHB 1058 was also isolated from us, but it was not mucoid. But if we look at the highly mucoid strain, and we look at lung homogenates or how long the animal survived, we see that there's a significant difference comparing this strain to all of the other strains. And if we look at the amount of capsule produced, we see that the typical lab strain, which we set at 100%, this CF 756 produced a far more capsule than that one did, and it also caused more disease and had a higher preponderance of death. If we look at the bronchial alveolar lavage, which is the bacteria sticking in the floating in the lungs as well as sticking in the lung, we still see that there's a significant difference. So yes, this does have a potential impact in cystic fibrosis. That's why these strains were coming out far more often than strains that appeared like this or this. And while I won't go any more to my biofilms, there's also a difference in a way. The large mucoid strains versus the small mucoid strains were able to make biofilms. Looking close to the lower level, we see a beautiful mixture here of live and dead cells. And that's the same thing here. But when you go to a less mucoid strain, we see that it's actually not the same. There are far more holes in this biofilm, and actually the biofilm is very unstable. So just to summarize, we saw serotype distribution differences. The CF lung actually may have something to do with why these mucoid organisms predominate. Something I did not get to talk about, which was originally going to be my topic, was S. pneumoniae competing out Pseudomonas. So when we're talking about novel antimicrobials, there's a novel system going on here that hasn't been described, where it's when bacteria kills off the other one. And that's where a lot of our original antibiotics came from. Using bacteria and flow cells, we were able to study the different types of bacteria that we saw. The highly mucoid serotype 3 caused more serious disease, and we were able to use our CFTR model to actually support this. And I want to acknowledge my lab members as well as the groups at UAB and Wake Forest University and other people who helped with this study. And I'll take any questions.
Time for questions. Started a little bit early,
So you mentioned earlier about the importance of mucus inhibiting the beating of the cilia, right? Mm-hmm. Maybe this is more of the biophysics approach, but how well known is it, sort of the molecular details of that mucus in terms of its viscosity and its components? Because can we prevent it from forming is one approach, or can we have some inhibitors to prevent it from reaching a critical viscosity so the beating isn't impaired?
Yes, it has been studied and characterized, and there are ways to prevent it. But it's when the CF patients have to put on their vest every day, I don't describe it well, but it's a shaking vest. And it makes this mucus not be able to stick. It'll make them cough it up. Shake, 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 or they do hitting therapies. Yeah. Yeah. Those therapies keep it from forming, physical therapies. Right. But there are also things along the line of albuterols that help it to not form as well. But are any of those 100% no?
So it's known that thinning the mucus,
lowering
the viscosity
yes.
to some extent.
Yes, it does have an effect on a positive effect on their overall health. Yes. How well it's been studied, I know that it's characterized, but I'm not sure what new therapies are coming in that area.
Yeah, so I remember you showed the slide with like the relative percentages of like the capsule material. Mm-hmm. Is it that material that contributes to the formation of the biofilm, or are they not really related?
Yes, it does contribute, but the short answer is yes. But there are other things, as many biofilms have a lot of components such as DNA and other polysaccharides that come as cells die and release into the environment. So yes, the capsule of polysaccharide does play a role, but even if it's absent, the bacteria will still form a biofilm. It will just be different.
in your images of biofilm, can you go to that slide?
Sure.
It looked like the early image on the left looked very homogeneous in its composition rather than green like randomly, and then the bottom one seemed to show that green ones are a little clustered.
Which one here?
Yeah. Is that real or that's just
No, it's real. So all of this is the same image.
Uh-huh.
If you look at this image here, it's at the base.
Uh-huh.
And this one is closer to the surface of the film. So when you get to the surface, you have this, where it's closer to the bottom, you have a mixture of live and dead.
So that is a known phenomena that they cluster like this. It's almost like phase separation.
For pneumococcal, yes.
And does the mucoid play a role? Does polysaccharide, or is it biological? So is it physical or biological? No Big Bang moment.
Like what causes the dead ones to cluster?
I think it could be that the dead ones, you know, you're going to have more limited nutrients at the bottom. Even if you have the challenge, you're not going to have the nutrients and the oxygen requirements and all closer to the base, because this is 48 hours old. So these would have been deposited much earlier. These are probably newer cells, because we were reaching probably close to the critical mass of how heavy this is going to get. And I think that probably has a, and you know.
So what was the thickness of that?
I don't remember. And I don't have my measurements on this
Yeah,
one.
I'm looking for the scale.
Yeah, I can't see the scale.
Okay. Yeah.
But that's going to influence it. And that's typical of biofilm. What's on the top is going to be alive. If there's nothing to impede it, they're going to be alive and usually fairly healthy cells, but they are part of the biofilm. So when your antimicrobials come in, this is all that's going to get killed, because they're growing. So when you go down lower, down to here, and you're trying to do things to kill the biofilm, most of these are dead, but they're still a part of the physical structure. So when you're talking about antimicrobials, you're talking about needing to remove this, and if the bacteria are metabolically inert, that's going to be harder to do.
Other questions? Yes.
So what's known strategies then for allowing the antibiotics to penetrate the biofilm? Because that's something we're very interested in, to have the entire biofilm attacked by the antibiotics. But if it's a skin layer and then below the surface,
I don't know that there are any good therapies for getting down to the lower levels. Most of it's going to sit on the surface. And remember, S. pneumoniae is gram-positive. So gram-positives are actually more susceptible than, say, an E. coli. Because what will happen is gram-positive, you can get the surface, and you may get some in the deeper, but with gram-negatives, there's actually usually pumps to push it back out. The antimicrobials will go in and they'll push it back out. So anything past this first couple of layers, there are no therapies for that. So learning how the biofilm, how the channels work, if the bacteria can make it into a channel and follow through a channel, possibly, but that's not easy to do.
So would you say then the therapeutic approach then would be more to focus on limiting the adhesion or the formation of that base layer to begin with?
Yes. If you can tap a property like
To release the whole thing.
the thin films that don't allow it to adhere in the beginning, if you can break up the adherence, you can prevent the biofilm from forming. Yes.
All right, with that, let's thank Mamie